Okay, so some announcements before we start. Progress reports are due tomorrow, and your student-led conferences are due tomorrow. I have a pile next to Damian, if you can turn it in today. We will continue our Unit 6 notes. You should have page 2 and part of page 3 completed. And your achieves are due today. Your coloring is due tomorrow. For Unit 6 assessments, you are going to have a quizzes. It's actually already posted. You may not feel ready to take it yet because you just started the unit, but your 6A quizzes will be due next Friday. I have given you the gift of unlimited chances, so I'm expecting everyone to get 100% on that quiz, okay? Your Unit 6 test will be combined. So your Unit 6 test will cover 6A and 6B, okay? And that will be on 10/18. So make sure you get your quizzes done for next week and then retain that information as we go through 6B. So in terms of collecting your notes, I will collect 6A. Actually, I may not be here on the 18th. My due date's on the 20th. So, I don't know. But your Unit 6A and 6B notes will both be collected on your 6th test. Okay, because I don't want to take your notes and then you need them to study. Cool. All right. Labeling the image to the right. I'll give you guys another minute to talk about that while I find my cards so I can call on you guys. Morning. Tests will be graded by tonight. We can go over those tomorrow. I don't remember. You ready? Actually, real quick. Who is the sun?
It's the whole monstrosity.
Actually, I would look it up, but they need that. All right. So from your notes yesterday, you should be able to label that image. Give you some hints in a moment. And a couple of you guys were late today. Let's try not to do that in the future. Yes, this image should be in your brain somewhere, for sure. Yep. And mitosis should be in your brain somewhere. Just got to pull it out. Pull it forward. All right. I'm going to shuffle. First person I call on, I'll give you the choice of which letter you want to give me. So you can give me whichever one you know best. If you did your homework, it's probably much easier than if you did not do your homework. All right, so we have A, B, C. What do we go up to? J? Oh, I get to teach the alphabet. I remember learning the alphabet. I really enjoyed it. I remember my teacher had these really cute pictures on the top of like her board. All right. Sammy, you are first. Which letter do you want to give me? B? E. Prophase. That sound good, Cassie? Yes. All right, good. All right, so that's kind of a hint to maybe some of the others, if you remember your mitosis. Mark. Any letter you want.
Honestly, I don't know.
Well, do you remember what comes after prophase and mitosis? Hemat. So M stands for what? Remember? All right, gotta do that homework. Oh, I can go over that in a minute. Kenya.
Metaphase.
Metaphase. Okay, good. Prophase, metaphase. So you should know, well, letter I will cover that. That's all, yeah. H would be, not quite one letter away. J is cytokinesis. Very good. Very close, very close. Okay. Sophia. We can go G. Yes. Prophase, metaphase. Anaphase. Good. Johnny? Interphase. Very good. Notice that interphase covers 3 different sections of our cell cycle. Okay, good. Benji. Telophase, very good. Telophase. So we had the acronym, oh, that was bad, PMAT. We could make a mnemonic for that. David, you had one. What was it?
Pass me a taco.
Pass me a taco. That's a new one. I haven't heard that one before.
Well, actually, this is
For
the-
your remembering the order, yes.
Don't I need to just pass me a taco? That's not okay.
Pass me a taco chip. Very good. But you do need to just make sure you know that cytokinesis is not part of mitosis. Yes, it comes right after, but it's technically a separate process. Okay, cool. Keep going. Alexis. Well, I is pointing to all of PMAT. So PMAT is representing the process of what? It is cell division. More specifically, it's division of the nucleus. Mitosis. Very good. Tanya? Gap 1, good. G1 phase. C is, or Dinar, you do C or D? Okay. S phase, good. And Ian? G1S then? You lost that homework? Go ahead, David. G2. Okay, and we're actually missing one thing on this picture that you did label in your notes. What is it? Anybody? Angel? You have mitosis?
Cell division. Yeah, cell division.
Well,
Wait, no.
this whole picture is cell cycle. No? On page 3. It's another G. Yes, G0. We're missing G0. Where on this picture would G0 go? Coming out of, is it on your picture? I think I want to do the label. We'll go over that. Okay, we'll go over G0. Cool. Thank you guys for being ready to be called on and participating. That was wonderful. So this is the cell cycle. You definitely need to know that picture. I would not be surprised if you had that picture or a very similar picture on a test where you would need to tell me what the pieces are, not only what the pieces are, but what happens in each of these pieces, which is what your notes were on last night. Okay. Okay, so you should watch this video at home. There's kind of a cool image showing you. This is showing you just the DNA throughout the cell cycle. So prophase in the yellow going through the stages, ending up with cytokinesis, telophase at the end. So here that is. So you want to make sure, okay, you should have done this for your notes, but you want to make sure you know what happens in G1, what happens in S phase, what happens in G2. We'll go into PMAT more specifically later in the notes. The next 2 that you needed to do, which is maybe why we didn't have Remember, it said also do slides 21 and 22. So 21, you got cytokinesis, which remember is separate from mitosis. It is not part of mitosis. And here is G naught. So if you could add to your picture in your notes, a little arrow of where G0 comes out. Okay, so it's after G2. So instead of going to, sorry, it's right after M. So after it divides, sometimes cells don't want to keep dividing. So right after M, right before G1, just put an arrow and put G0. Let me go back to your other picture. This picture looks more like your picture, right? Okay, so from this picture, you would take right where, see how this arrow takes you to B? Put another arrow coming off of that, or wherever that is on your picture, and put G0. You can think of G0 or G naught as like a resting phase. That cell is no longer going to prepare to divide. It's just gonna be doing its job for the rest of its life. Like the notes say, sometimes cells in G naught do go back into cell division. Sometimes they don't. It just depends on the cell and on what your body needs. Okay, so you're just adding a little G naught to your picture. All right. So we'll start with this question. Mitosis is the division of the nucleus into two genetically identical daughter nuclei. Any reasons why we need to do this? Why is this important? Why do we need to divide the nucleus? Yes. To get more, because this, yes, why? Why do we need more? Yes. Reproduction is one thing, yes. So if we skim forward to meiosis, sexual reproduction, we also do PMAT, right? You just do it twice. So to make our sperm and egg, yes, we need to have extra DNA. But not only that type of reproduction, remember, we also do asexual reproduction. For what reason? Why would we make a clone of our certain cells? To keep us alive, yes. Or specifically, our cells are constantly dying, and we need to constantly regenerate them, okay? It's like your hair keeps growing, right? And you keep making more hair. Same thing inside your body. Your skin cells are constantly dying and coming off. That's why your tan doesn't live forever, right? It goes away. Some of you guys were born tan and it's beautiful. But your cells constantly, even in your gut, like all in your body, your cells are constantly dying. You need to replace them. Or even wounds, right? We'll get into that. So here are some of the reasons. So in your notes, you're on page, what page? Yes. The role of mitosis. So there's basically 4 reasons here of why mitosis is important. So during embryonic development. So you can see an image here. Oh, that's Zachary's life. Actually, he's 37 weeks yesterday. It's getting so old. Me too. That's crazy. He's not even born yet, and I feel like he's already old. So the hundreds of millions of cells that make up the bone and flesh of your body are products of thousands of generations of cell division that began when you were smaller than the period at the end of the sentence, like microscopic small. Started when a single cell cleaved into 2 parts, then quickly reorganized and split again into 4 new cells. 4 cells became 8, 8 became 16, exponential growth pattern. The cascade continued until several weeks later, millions of cells were dividing, following the exponential pattern. So yeah, that's embryonic development. If you need an example, I would say, you could just say human baby. Human going from egg to blastocyst to fetus to baby. You guys know what a blastocyst is from last unit. Yeah, that happens. Me too. You can have that one if you want. Keep it. I have a whole. I actually ordered a new pack. Okay, that example is pretty self-explanatory, but of course, embryonic development can happen in many different types of animals, right? Here's a video of that.
Looking into the ovary at an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here are millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus. And then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring. And this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days.
Remember, what do we get from a blastocyst?
At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there
From last unit. Stem cells, specifically which kind? Pluripotent. And coming from where in the blastocyst? The inner cell mass. Very good.
are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc. And then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo.
It either is already
Those
living or
lines
it has the
represent
potential
sites
to
where
live,
cells
and
are
you stopped that
migrating in and
potential.
out. And here's an important stage when the three beginning layers of the embryo, the so-called germ layers.
So yeah,
are
you
formed,
did
and
used
I'll
to
come
look
back
like
to
that.
in a few minutes.
Look at you, you're so
As
cute.
development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns and appendages start to bud out. You see the head forming and the eye. And then eventually we get a
We do.
small
We start with
embryo,
the tail, and
and
the
some
tail goes
months
away.
later, of course, this would be born as a young baby.
Yeah, and actually some humans are actually born with tails. Like little tails. They're usually like about this long. No, they are humans. You never see it because most doctors are like, do you want us to remove the tail? And most parents are like, yes, please remove the tail. There are babies who, especially in other countries, they do keep the tail because they believe it's a sign of them being special, or they just didn't have the money and the resources. But you can look it up. It looks weird, and my students are never mature enough to handle it, but you could look up, maybe not on school Chromebooks, but babies with tails, you can see they do exist. Yes. Yes.
In this video then,
Organism growth. This one you have multiple examples you could choose. You could choose to say baby tarantula to adult tarantula. You could say plants growing from a seed to a tree, or seed to a bush, or any organism. Pick your favorite organism and talk about how it can grow, right? I actually have that exact tarantula because we used to have a tarantula. We used to have 8 tarantulas at our max. 8. We just kept buying them. We had all different kinds. 8th grade, I had 1. Yeah. Some of them that come from areas not near here are hard to keep alive in this area. You could just pick a human growing from baby to adult, or plant growing from seed to flower, something, anything growing. Probably not till my babies are grown. We have 1 tarantula at home, but it's the least scary tarantula. The least dangerous. We used to have more dangerous. We used to have centipede that could cause a lot of damage. We used to have ones that hurt. We have the ball python at home still. My mom wishes I would bring him here because she doesn't like the idea of my baby living in the same house as a snake. The snake is in the office and the baby will never go in the office. Here's something growing. Whee! This is an avocado pit and they have it suspended. One of my students said she actually tried to do this and it was growing, but it took like forever. She said it was actually more frustrating because it took so long for it to start actually sprouting and growing. Look, there's a little root down there. Yes. Yeah. Nice. How long did it take to get there? Yeah. My sister-in-law, she was eating bell pepper one day and she saved the seeds and planted them. And now that bush is finally making bell peppers, which is fun. I don't know. She's also growing other things too. But, all right, this could be forever. Yeah. Then it starts coming out the top. So what we're watching is mitosis in action, right? Cell division in action. I see a lot of people writing. Let's not go forward. And if you didn't do your homework, you got to do that at home. Whee, it's finally turning into a tree. Yep, and you probably need to keep, I think at this point you need to keep it under the pit because you don't want the pit to get moldy. And you need to keep refreshing the water. Not yet. And you also, you could do never soil if you just give it nutrients in the water. Why is it dancing? It's changing with the sun. It's called phototropism. All right, cool. Mitosis happens. Yay, growth. Tissue repair, that's your next one. Sorry for the disgusting picture. You can use, if you don't like that example of human wound, you could say, for example, Leo. Leo, you guys saw his tail is growing back from being attacked by a cat. Well, he lived with my sister for a while and she had the lizard in her office because the cat wasn't supposed to be allowed in the office. But her 5-year-old at the time was like, let's see if the cat and the lizard want to play. And so she didn't. She actually, so the lizard was still in the cage, but the cat jumped on the cage and was like clawing at it and trying to get at it. So he actually ditched his tail to run away, which is his defense mechanism. And so he like ran into his little hide. And then my sister was like, oh, the cat is being crazy, and then went and got the cat. But that's why he ditched his tail. Cockroaches? I think a lot of insects will do that just because their nervous system is still firing. Humans do that too. We twitch after we die and stuff. It's weird. Okay. Here's another video. How do wounds heal? Well, mitosis, but there's more to the story. So one of the things I've noticed that there is a correlation to is students who are doing notes while we're doing lecture don't generally do as well on tests. Okay, so sometimes it's because they're behind, sometimes it's because they're trying to go forward. All of that needs to happen like on your own time. Okay, so if I'm showing you a video, you should be only watching that video. If I'm discussing something, the reason I actually go over these notes is because us talking about it helps your brain understand it. So if you're just taking notes and I'm just like talking and you're not listening, you're not getting the full educational experience. Okay, so I'm feeling like I don't know if we need to have a rule of no Chromebooks when we're doing lecture, but let's just make sure we're not doing other things that are not science. during the time that we're doing science, or trying to catch up or going ahead in our notes. If you're catching up or going ahead, that needs to happen on your own time.
Skin, the surface area of about 20 square feet in adults. Though different areas of the skin have different characteristics, much of this surface performs similar functions, such as sweating, feeling heat and cold, and growing hair. But after a deep cut or wound, the newly healed skin will look different from the surrounding area and may not fully regain all its abilities for a while, or at all. To understand why this happens, we need to look at the structure of the human skin. The top layer, called the epidermis, consists mostly of hardened cells, called keratinocytes, and provides protection. Since its outer layer is constantly being shed and renewed, it's pretty easy to repair. But sometimes a wound penetrates into the dermis, which contains blood vessels and the various glands and nerve endings that enable the skin's many functions. And when that happens, it triggers the 4 overlapping stages of the regenerative process. The first stage, hemostasis, is the skin's response to 2 immediate threats: that you're now losing blood and that the physical barrier of the epidermis has been compromised. As the blood vessels tighten to minimize the bleeding in a process known as vasoconstriction, both threats are averted by forming a blood clot. A special protein known as fibrin forms crosslinks on the top of the skin, preventing blood from flowing out and bacteria or pathogens from getting in. After about 3 hours of this, the skin begins to turn red, signaling the next stage, inflammation. With bleeding under control and the barrier secured, the body sends special cells to fight any pathogens that may have gotten through. Among the most important of these are white blood cells, known as macrophages, which devour bacteria and damaged tissue through a process known as phagocytosis, in addition to producing growth factors to spur healing. And because these tiny soldiers need to travel through the blood to get to the wound site, the previously constricted blood vessels now expand in a process called vasodilation. About 2 to 3 days after the wound, the proliferative stage occurs, when fibroblast cells begin to enter the wound. In the process of collagen deposition, they produce a fibrous protein called collagen in the wound site, forming connective skin tissue to replace the fibrin from before. As epidermal cells divide to reform the outer layer of skin, the dermis contracts to close the wound.
So that's the part right there where the cell division is happening, right? So all of these cells that are doing all of these jobs, those cells don't necessarily live forever. So those cells have to constantly be regenerated. And some of them only get made when your body is like, I need this cell. So then it starts to make a bunch of those cells. And then also just to repair the tissue, right? If you have a cut, you don't have a cut forever, eventually seals up. And that's because you're making new cells to do the sealing. Yes. Well, a scar is basically just a different type of tissue that was filled in. It's called scar tissue. So it's a slightly different composition and makeup than your skin that you had before. I don't think so. I think it's just newer. And what you'll notice too, like with certain scars that aren't super deep, they actually will fade over time. Like I have a scar on my arm right here that used to be really, really prominent. And now it's fading. A couple more years, you probably won't even notice it. So eventually, even that tissue, that scar tissue will move on and be replaced by, because that's like fast tissue, right? Like your body is like, I need to seal this cut right away. It's not going to have time to do the slow process. So over time, as the scar tissue sloughs off, just like all the rest of your skin, then you're being replaced with like the normal skin growth. But some scars are super deep and they never go away. And like tattoos too. Depends on how deep in the layers of skin it goes.
As epidermal cells divide, newly deposited collagen is rearranged and converted into specific types. Through this process, which can take over a year, the tensile strength of the new skin is improved, and blood vessels and other connections are strengthened. With time, the new tissue can reach from 50 to 80% of some of its original healthy function, depending on the severity of the initial wound and on the function itself. But because the skin does not fully recover, scarring continues to be a major clinical issue for doctors around the world. And even though researchers have made significant strides in understanding the healing process, many fundamental mysteries remain unresolved. For instance, do fibroblast cells arrive from the blood vessel?
Okay, we gotta move on. Bruises are basically just you're bleeding on the inside. And that's why you see that discoloration. And then eventually that all kind of spreads out and goes away. Yeah. Why are we still doing notes? It's called a contusion. Basically, you rupture blood vessels on the inside of your skin without cutting the outside. Yeah. And sometimes, like my friend, she hurt her leg so bad she had an ATV fall on her leg because she didn't know how to drive it. She tried to go up a hill and then tried to turn down the hill and it just toppled on her. But her bruise was so bad that it just kept bleeding and kept bleeding. And so her skin, like inside of her leg, was swelling up. So they had to release the pressure. So when she went to the hospital, they basically had to cut it open and let all the blood come out, or else her skin would have like exploded. So that was not good. Then it got infected. It was a whole thing. No. The problem was she didn't have insurance, and that she didn't go to the hospital when she should have, which caused problems. She just wasn't being responsible in life at the time. Here's this marker growth. This is Leo. This is what his tail looked like when he dropped it. And then it started growing back. Yeah, that's a very interesting thing. It's like, how would you just be like, drop your arm? Like this. I don't know. You would have to ask a bird because those are usually the ones that eat it. So that's kind of where he's at now. And eventually, over time, it never goes back to normal. It'll always be kind of like this bulb shape. All right, so hopefully you have your example of tissue repair. The last one is, yes, asexual reproduction. So you guys already kind of looked at this in your last set of notes. So for your example, you could do developing bud, okay? I would just put budding, okay? Budding is a process that happens in yeast cells and in this hydra. You can either put budding in a yeast cell or budding in a hydra. That is a form of asexual reproduction where the organism just starts cloning its cells and then it keeps growing and growing and then it cuts off to become its own organism. So these are all reasons why mitosis needs to happen. Okay, so we're making more cells, but remember the question goes back to why mitosis? So mitosis, this is a common misconception. Mitosis is the division of what? Of the nucleus, exactly. Okay, when somebody asks you what is mitosis, you don't want to say cell division. Okay, cell division is mitosis. Cell division would be mitosis plus what? Cytokinesis. Okay. So the question is, why do we have to do mitosis? Well, because if we want to make two cells, we need two nuclei for that, right? So first we have to divide the nuclei so that eventually in our two cells, they both have identical DNA. Okay. Fair question? Here's some asexual reproduction. Actually, oh, fun. Amazing. We'll see if it's good. Gosh, 5 minutes. We may not do all 5 minutes. But you could also use these for examples too. So binary fission happening there. You guys know that from unit 3. They're eventually going to be separate. Almost. Come on, you can do it. Oh, wait. Yay, they're finally separate. Okay. Number 2 is spore propagation. You can put that in as an example too. Here's bread mold growing. Yeah. Don't let your mold get that big. That's dangerous. This is how mold spreads. The spores, you can kind of think of them almost like pollen, where they kind of just like break out and spread everywhere. Spores is specifically the dangerous part of mold. You can be sick from mold, right? And it's usually from ingesting the spores. Flag mold? Just a specific species. I don't know the species. Number 3, budding. Oh, here's the hydra. Bloop, little baby hydra. Genetically identical, then it will start to grow on its own. Whee. Oh, and regeneration, that's another one you can put as an example. Poor starfish got an arm cut off. But look, it's regrowing. Good job, starfish. Cool. Wasn't a terrible video. I like it. Yeah. In the very middle is like where their like stomach is, and they kind of just like suck things. They're a very important part of the ecosystem because they are food for other things, and they eat the smaller things. Because in populations, we don't want things to dominate or overgrow. So they will eat some of the smaller organisms so that the smaller organisms don't take over. Yes. No. Binary fission is actually very similar to mitosis because it's just one cell and it just grows and then divides. Yeah. Okay. I think this might be out of order, but I think this is a really good link. Like I said in red, you should actually look at the link because it's going to take you deeper into exactly what's happening during DNA, during mitosis. So we'll look at that when we do some of our drawings, but I read through this and I feel like the way it's written is really helpful. Okay. So highly, highly suggested to read if you want to do well on a test. Okay, so we already went over that. I think I want you guys to get online and do this. So you can get on your Chromebook, and you guys, I just want you to go through this on your own and review the topic of the cell cycle. So you're gonna go into your Unit 6A presentation, and you're gonna be under slide 23. And 24 will be for homework. I want you to watch that video for homework. Okay? So you've got, what, 10 minutes? You've got 10 minutes to do the review. Now could be a time where if you need to get caught up on notes or you have a question. Oh, somebody asked me about the coloring sheet. So to review the mitosis close read and coloring page. Notice that I have put an image of the colors, so make sure you are using exactly those colors. Okay, and what I need you to do is close read the front. This is important information. Okay, it's going to tell you now. I would do the coloring as you're reading because look, it says Read this. Now color interface, color the heading interface, then read this. Color the heading prophase, now read this, okay? So as you're reading the front, you should be coloring the back kind of in that order. Close read, what are we doing in close reading? Annotating. Do we also highlight something?
Yeah.
Highlight. Some of you guys underline instead, which I guess if it works for you. I like highlighting better, but that works for you. And then just color it. So that's pretty much all you need to do. When is this due? Tomorrow. When are your achieves due? Today. Cool. All right, so you can have 10 minutes to do your review and then write down somewhere that you're gonna need to watch the video on slide 24 for homework. Okay, all right. I'm gonna turn the light on. Yes. Yep. Well, 2 each, yeah.